All right, I'm going to quickly go through the origins of life and our evidence of early Earth and some of the dates that you'd need to know. When we talk about origins of life and just when we talk about origins of different organism groups as we go through the semester, we're thinking on what we call deep time, which is sort of a difficult concept for us as humans. You know, we live in a kind of time frame where 100 years is really, really long. Or 2,000 years is long. But in the scale of the history of the Earth, it's a very, very, very short period of time. Just to kind of give you a perspective, to remind you what a billion is, 1 billion is 1,000 million. And here are some kind of conversion. So a billion seconds is 31.7 years. So if you have, someone who's 30 years old, they're a billion seconds old about. To spend $1 billion per year, you'd have to spend over $2.7 million a day. So you can see a billion is a very large number. So just keep that in mind when we're talking about this kind of scale. One of the numbers that you do need to know is the date of the Earth formation. So that's about 4.6 billion years ago. And I'm not going to go through the evidence for that in particular, but I can direct you to some resources if you would like. So 4.6 billion years, and then for about a billion years, we don't see fossil evidence of life yet. So this is the first type of fossil evidence we see in 3.5 billion years. These structures here that are called stromatolites. They're formed by, we have them current day as well, they're formed by bacteria putting down layers of deposits, basically. And so they end up forming these little stepping stone looking things. And so we know for sure 3.5 billion years ago life existed and it was prokaryotic. We have good chemical evidence that leads us to currently pretty much the consensus is about 3.9, maybe 3.8 billion years ago. And I can show you time. So here's the formation of the Earth. 4.6 approximately, and this million years, of course, so that's 4.6 billion years. And the Earth basically is this fireball and then forms rock, and then right after that is when we start to see this sort of thing. Here's the chemical evidence.
Bacteria are small, but they are so numerous that they can actually influence the chemical composition of seawater. What this cartoon is meant to illustrate is that in the process of photosynthesis, cyanobacteria or any other photosynthetic organism will have a menu, if you will, of CO2, some of which contains the heavier isotope of carbon, heavier stable isotope, carbon-13, some of which contains carbon-12. And for reasons that simply have to do with the differing mass of those CO2 molecules, the C12-bearing CO2 will be preferentially incorporated into organic matter doing photosynthesis. Now what that means is that if we can measure the ratio of C13 to C12 in the organic matter and then measure the same ratio in limestones associated with that organic matter, they will differ in that ratio. They'll actually differ by around 25 parts per thousand. We can measure that with a mass spectrometer, and we do that for the Spitsbergen rocks. We have strong confidence that there was a biological carbon cycle driven by photosynthesis on the basis of that chemistry.
The history of microbial life extends very far back in time, as far as 3.8 billion years, as we currently estimate, but this might have been even earlier for all we know. How do we decipher when particular microbial metabolisms evolved and what types they were? Well, this indeed is extremely challenging. And there are three primary ways we can gain insight into the microbiology of the past: through using either morphological, molecular, or genomic, which is, of course, a form of molecular, biosignatures. And these are very different in what they can tell us. So the first two, morphological and molecular, are important because they can be concretely linked to rocks, old rocks that we can date. And because of this, when we see a particular form, this is being held in a hand, a sample of a stromatolite. This is at a very different scale. Here, you're looking at a thin section of a rock, and that's true for these images below, where the scale is about 1 millimeter in this image, and it's even smaller down here, the structures that you observe have been interpreted as being vestiges of ancient life for various reasons. But this interpretation is often ambiguous, and it's a challenge to be able to come up with unambiguous biosignatures simply on the basis of their shape. And so geobiologists, those interested in seeking to understand life in ancient times, have turned recently to what we call molecular biosignatures that come in two forms, either organic biosignatures or some type of inorganic biosignature, often expressed as a ratio of different isotopes in a sample. Now, this in turn is challenging as well, but it may be the best way that we can gain more specific insight into different types of metabolisms by looking at actually the chemistry of what's left in the rock and being able to deduce through finer scale analyses whether or not this chemistry is one that was uniquely imparted by a biological process. Lastly, we can think of genes as fossils, and the genomic record has been crucial in establishing the diversity of life on the planet, as I'll get to in a little while in this lecture, but it also helps us understand the relatedness of different enzymatic functions and how they evolved from one another. While this does not give us a concrete date when these metabolisms evolved, it does provide us with an ability to look at the relationship between different metabolisms and come up with an order in which they likely were invented.
Okay, so you can see this other timeline here, and here's a nice close-up of what a stromatolite looks like, and here's modern-day stromatolites as well, just to show you that. Okay, I like this poster here. It's actually a large poster. I don't have it with us now, but it shows the geology and correlates that with the biology that's going on in the earth at the time. It has the phylogenetic tree in there as well. You can see the mitochondria and the chloroplast symbiosis events, endosymbiosis. There, what you'll notice here is that, okay, here's the formation, there's liquid water, there's the rock, here's the chemical signatures we just saw, then here's the stromatolites, 4.5. Homo sapiens, that's not even modern day humans, that's just the genus that we're part of. We're here. So even dinosaurs are right there. So we're talking a long time ago. We don't have a lot of oxygen in this early environment, and that's going to be important because it likely contributed to how the first cells could form, and really how the first molecules could form from smaller components. I don't need you for our class to know these names of the eons, but I do need you to know the basic order that things happen in. I told you 2 dates already, or 3 I suppose, the 4.5, the 3.8 we'll say, and then 3.5 being the definitive fossil evidence of those prokaryotes. And then we don't actually see the eukaryotes until 1.8 or so billion years. So that's a billion years, maybe 2 billion years really. If it's 3.8 to 2.8 to 1.8, 2 billion years that are just prokaryotic life. And then you'll actually notice there's no multicellular organisms all the way until probably about 1.5 or a little bit less than that. Because everything here is pretty much in water. And you can also see atmospheric oxygen starts to build somewhere around 2.5 billion years ago. We call that event the oxygen revolution, you can see here. And that really changed the landscape, or the atmosphere, I should say, which changed the microbial landscape of the world. In low oxygen conditions, so anaerobes, which are organisms that die in oxygen, if they're obligate anaerobes, then they would go extinct when the oxygen levels went up. Other organisms that use oxygen could expand, and so this would actually be an example of where you'd have a mass extinction going on, you'd have adaptive radiation happening, so that would be one of those kinds of situations where you'd expect a lot of expansion of diversity very quickly, relatively quickly in deep time. So how do we know that the oxygen levels were low, and how do we see when they went up? And when they went up, that's telling us where, photosynthesis really started to kick in. And since we don't have eukaryotes, that's definitely cyanobacteria that's doing the photosynthesis. So this again is where we use the geological rocks and look at their mineral components, and this is going to tell us about oxygen levels.
This is iron formation, and again, I have an example here. Iron formation is a rock that's commonly banded, and the bands contain iron minerals. The red here is an iron oxide hematite, and much of the iron that has ever been mined for industrial purposes formed on the early Earth as these deposits. Now, the interesting thing about iron formation is it cannot form in the oceans today, not even in principle. And the reason is that when you put iron in a solution that contains oxygen, it immediately precipitates as an oxide. The evidence of these sedimentary rocks is that that iron was actually transported through deep oceans, perhaps for long distances, without ever coming in contact with oxygen. Ferrous iron, or reduced iron, can travel in solution very well.
Okay, so this is a little animation showing some hypotheses about how oxygen levels actually started to rise, because you can produce oxygen with photosynthesis, oxygenic photosynthesis is what they call it. But unless the balance between oxygen consumption and oxygen production is tipped, if it's completely balanced, the oxygen won't accumulate still. So how might that happen? Okay, here's the imbalance of photosynthesis and cell respiration. So as you know, photosynthesis is taking CO2 and H2O, electron donor, water, and oxygen, or carbon donor, carbon dioxide. Photosynthesis turns that into organic matter and then oxygen. 2.7 billion years ago, cyanobacteria are doing this. But remember, cyanobacteria, as well as other organisms, all produce ATP, which remember is not the job of photosynthesis. That's the job of respiration or fermentation. Now, fermentation is a type of production of ATP that does not use oxygen, so that would not be contributing here to this backwards reaction. Anaerobic respiration does not use oxygen. Some organisms do that. That would not be contributing. But any aerobic respiration that uses oxygen as a starting product in order to break down basically the organic matter, which ends up releasing water and carbon dioxide and producing ATP. The cyanobacteria themselves are doing that, and the other aerobically respirating prokaryotes that exist at the time as well. So here we have our oxygen consumption and production in equilibrium. So we have steady low levels of oxygen. But if we look, there's an idea that we have this organic matter, which is basically the product of photosynthesis. So that's, in my class, I call them carbon compounds. They would become buried in sediments. And so since you don't have the organic matter to go backwards in cellular respiration, so basically those organisms die, if you want to think of it that way, goes away, then you won't have them going backwards. And so therefore you will not have as much cellular respiration, and so the oxygen will accumulate. Another hypothesis here is looking at oxygen kind of getting used up and not. Let's look at this way. Okay, so oxidation is the loss of electrons. Oxygen is going to be able to pick up electrons. Remember, it's a big electron sink. So it's highly electronegative, the oxygen atom, and oxygen can grab those electrons and take a reduced compound to an oxidized compound. If the reduced compounds were depleted, as in the iron 2, once it's all been turned to iron 3, then the oxygen would accumulate because there wouldn't be reacting with those reduced compounds to oxidize them, because there wouldn't be any more of those. So that's another way the oxygen levels could rise. Then another thing I want to show you, here's that great oxygenation event, kind of like what we were talking about, accumulation here. Before that, and one of the videos had mentioned about looking at biomarkers, biological molecules. So here is an example of that.
And there are still other ways that microbial life leaves a signature in sedimentary rocks. Most sedimentary rocks, including the ones from Spitsbergen, have at least a little bit of organic matter that survived decay and was incorporated into those rocks. They tend not to contain things like DNA and proteins. Those are too easily broken down by bacteria in search of food. But things like lipids preserve pretty well. So one thing to think about is that when you die, the last bit of you to be present for future generations to ponder might well be the cholesterol in your blood vessels. So think of that next time you have a hamburger.
So thinking about these low oxygen conditions and other kinds of molecules in the atmosphere, when we think about that, and I'll show you those in a moment, think those conditions, and they tested a variety of different conditions because, we don't know exactly what was in the atmosphere. They definitely do research on that for sure. We think that those can make the origins of life possible. So here's kind of the way that we state the 4 basic steps.
What we're trying to do is really come up with a plausibly realistic sequence of events so that we understand all of the transitions throughout this whole pathway. And we'd like to understand a complete pathway from planet formation through early chemistry, more complicated organic chemistry, up to the assembly of those building blocks into the first cells, the emergence of Darwinian evolution, and then the gradual complexification of early life leading up to what we see now. Okay, so let's look a little bit more closely at the chemical steps. So in broad outline, what we think happened is that you start off with very simple molecules, such as shown up here. There's still a lot of debate about the nature of the early atmosphere. Scientific opinions have gone back and forth in terms of the structure and how reducing that atmosphere was. But it's also been recognized that there could be very important local variations. So even if the atmosphere was globally fairly neutral or perhaps mildly reducing or mildly oxidizing, there could be local environments that were more reducing. That, together with the input of various forms of energy, for example, from electric discharges, lightning, high energy ultraviolet radiation, ionizing radiation. These are all forms of very energetic processes that can basically rip these small starting molecules apart into atoms, which can then recombine to generate high energy intermediates with multiple bonds. Molecules like cyanide and acetylene, formaldehyde, and so on. And these molecules can then start to interact with each other and gradually build up more complex intermediates, ultimately leading to the things we really care about, the lipids that will make membranes and vesicles, the nucleotides that will assemble into genetic molecules like RNA, amino acids that can assemble into peptides, which may also play roles in primitive cells. And somehow, and this is the question that my lab has really been focused on, somehow all of these molecules come together and assemble into larger structures that look and act like cells that can grow and divide.
One of the most famous experiments in science was done by Stanley Miller when he was in Miller-Urey's lab. It was in 1953, around the time when DNA was structured, that era when that was being discovered. And what they basically showed is that you can make carbon-based organic molecules from simple gases with water vapor and with an addition of energy. In this case, it was like a spark, which represented lightning. So let me show you the Miller-Urey experiment. It's been repeated with many, many different conditions since then, and has shown that we can make a bunch of different types of molecules from that.
Stated Clearly presents What was the Miller-Urey experiment? It was once believed that if you left food out to rot, living creatures like maggots and even rats would simply poof into existence. The idea was called spontaneous generation. A series of experiments starting in the 1600s disproved this idea, and in the 1800s, a new scientific law was proposed. Life only comes from life. It's true that rats, maggots, and even microbes are far too complex to simply poof into existence, but in 1859, English naturalist Charles Darwin put forth the theory of evolution. In it, he showed that under the right circumstances, relatively simple creatures can gradually give rise to more complex creatures. Given this information, serious thinkers began to wonder, is it possible that simple life forms actually could come from non-living matter? Not by poofing into existence, but through a natural gradual process similar to what we see in biological evolution. Darwin himself mentioned this idea when writing to a friend. But if, and oh what a big if, he wrote, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, and so on present, that a protein compound was chemically formed, ready to undergo still more complex changes. In 1924, Russian biochemist Alexander Oparin published a book which he titled The Origin of Life. In it, he outlined his thoughts on a gradual progression from simple chemistry to living cells. He imagined the early ocean as a primordial soup, a rich collection of complex molecules produced by natural chemical reactions. In this soup, further reactions could take place, eventually producing living cells. At the time, Darwin's warm little pond and Oparin's primordial soup were really just speculation. They were founded on a good understanding of chemistry and biology, but they could not be considered legitimate scientific hypotheses because no one had found a way to test or observe them. Science, after all, is the study of observable facts and an ongoing conversation about how those facts can be best linked together. Chemical reactions like those proposed by Darwin and Oparin are not expected to leave an observable fossil record. Without either having fossils to examine or a time machine to travel back and observe what happened, how could scientists even begin to study the origin of life? In the 1950s, Stanley Miller, then a graduate student at the University of Chicago, came up with an idea. We could simulate early Earth conditions in the lab and then carefully watch what happens. If you can't study fish in the sea, set up an aquarium. Working with his professor, Miller-Urey, Miller designed an apparatus to simulate the ancient water cycle. Together they put in water to model the ancient ocean. It was gently boiled to mimic evaporation. Along with water vapor, for gases of the atmosphere they chose methane, hydrogen, and ammonia. These are simple gases which scientists at the time thought were probably abundant on the ancient Earth. They added a condenser to cool the atmosphere, allowing water molecules to form drops and fall back into their ocean like rain. The ancient Earth would have had many sources of energy, sunlight, geothermal heat, and even thunderstorms, so they added sparks to the atmosphere to simulate lightning. The goal of the experiment was not to create life, but to simply test the first step in Oparin's model. Can simple chemistry naturally give rise to the complex molecules of life? After running the experiment for just 1 week, their ocean became brownish black. Careful analysis revealed that through a series of reactions, many complex molecules had been produced. Among these were amino acids, special molecules of life that we once thought could only be built inside the bodies of living creatures. This was a pivotal breakthrough in science, so significant in fact that it gave rise to an entirely new field of research now known as prebiotic chemistry. Scientists don't know for sure if the gases used by Miller really were the most common gases of the ancient Earth. Because of this, many experiments have since been done, showing that the molecules of life can form in a wide variety of environments with different starting chemicals and different sources of energy. Sugars, lipids, and amino acids have even been found on meteorites. This suggests that the molecules of life formed all throughout the ancient solar system and may be forming right now in other regions of our galaxy. Together, these discoveries tell us that Oparin's primordial soup and Darwin's warm little pond could have easily existed in one way or another on our ancient planet. So to sum things up, what was the Miller-Urey experiment? The Miller-Urey experiment was our first attempt at simulating ancient Earth conditions, in this case, the ancient Earth's water cycle, for the purpose of testing ideas about the origin of life. The Miller-Urey experiment is significant for two main reasons. First, though it was not a perfect simulation of the early Earth, it clearly demonstrated for the first time that biomolecules can form under ancient Earth-like conditions. Second, the experiment took what was once mere speculation, the idea that life may have emerged from chemistry, and transformed a portion of that speculation into legitimate, testable science. Many questions remain to be answered about the origin of life, but scientists from many nations and many fields of study are now following Stanley Miller's lead. They're finding ways to turn those questions about the origin of life into testable, scientific hypotheses. Simulation experiments cannot tell us exactly how life formed in the past, but if enough of them are done, they could eventually tell us if it's possible for life to emerge from chemistry. I'm John Perry, and that's the Miller-Urey experiment, stated clearly. This video was funded by the Center for Chemical Evolution, the National Science Foundation, and NASA. Special thanks to chemist Eric Parker. He volunteered hours of his time going over our script, sending us scientific papers, and critiquing our visuals for this animation. Though we do receive grants from time to time, Stated Clearly is made possible with financial contributions from viewers like you. To support us,
I'd also just like to show you that there is such a thing as volcanic lightning, in case you're unaware of that. There's a lot of volcanic activity on the early Earth, we know that. And so actually you can see it here. And it's generated just because of charge differences within the cloud of ash and the surface. You see the flash. There you can see that. So that actually is an energy source that they think may have been a place that could contribute to this abiotic synthesis of these small organic molecules. Okay, now we're going to look at taking those small organic molecules and moving or building them up into larger macromolecules, things like RNA molecules instead of just RNA nucleotides. So I'm just going to start with one of the things that for early life is building nucleic acids. And here's one example of how that may have happened.
Researchers have shown that clays, such as montmorillonite, are able to catalyze the formation of polymers of RNA from single nucleotides. Nucleotides, shown in white and blue, adsorb to the surface of a small clay particle, shown in brown. As the surface becomes more crowded, nucleotides that are close together can undergo a chemical reaction resulting in the formation of a polymer, or strand, of RNA. Hydrothermal vents, from deep-sea black smokers to land-bound geysers, may have been sites where prebiotically important molecules on early Earth were formed. This animation shows the formation of fatty acids deep in the earth below a geyser. Mineral surfaces can catalyze the stepwise formation of hydrocarbon chains from carbon monoxide and hydrogen. Here hydrogen atoms are shown as white spheres, carbon is gray, and oxygen in red. The reaction results in the growth of hydrocarbon chains of various lengths that are eventually released from the mineral face as fatty acids and related compounds. Because the fatty acids are at low concentrations in the water, they are unable to form higher order structures such as micelles and membranes. Following the violent explosion of the geyser, some water is released into the atmosphere as tiny, microscopic droplets. Fatty acids synthesized along the mineral walls of the geyser are found in low concentration in these droplets, with the longer fatty acids at the air-water interface. A gust of wind evaporates the water molecules in the water droplet, causing the fatty acid to form lightweight airborne particulates that can be transported across the landscape, perhaps eventually settling out and accumulating in localized areas. Under the proper conditions of concentration and pH, fatty acid micelles may join together to form a vesicle. Individual micelles first join to create a large, sheet-like bilayer. Random fluctuations of the fatty acid bilayer lead to the formation of a cup-like shape. When in close proximity, the ends of the sheet can seal together, forming a spherical vesicle. Recent research has shown that single nucleotides of RNA are able to traverse fatty acid vesicle membranes. This animation shows one mechanism by which nucleotides may enter the vesicle. Nucleotides bump into and interact with fatty acid head groups, colored in red, in the outer leaflet. Some of the fatty acids interacting with the nucleotide flip to the inner leaflet, carrying the nucleotide along and releasing it on the inside of the vesicle. When fatty acid micelles are added to a solution containing preformed vesicles, the vesicles are observed to grow rapidly. This is thought to happen through the formation of a micelle shell around the vesicle. Individual fatty acid molecules are then transferred from the micelle to the outer leaflet of the vesicle, resulting in vesicle growth and micelle shrinkage. Fatty acids must flip-flop between the inner and outer leaflet of the vesicle to ensure even growth of the vesicle membrane.
Stated Clearly presents What is the RNA World Hypothesis? If you were to go back in time 120 million years, you'd find yourself in a dinosaur world. 500 million years ago was a world of trilobites and other strange sea creatures. 3.4 billion years ago was the world of the first living cells. And if you were to go back further still, scientists suspect that chains of a chemical called RNA, or something similar to RNA, kickstarted this entire beautiful mess that we call life. RNA is thought to have given rise to life for several reasons. Chains of RNA are found abundantly in all living cells today. RNA is a close chemical cousin to DNA, and with very little help from researchers, RNA chains can replicate, evolve, and interact with their environments. While many details have yet to be worked out, the RNA world hypothesis is the simple idea that somewhere on our early planet, perhaps in a tide pool or hot spring, the Earth's chemistry was producing random chains of RNA. Once formed, they began replicating, evolving, and competing with each other for survival. As these chains evolved and diversified, some eventually began cooperating to produce the genetic code, a wide array of complex proteins, and even living cells which, from the perspective of RNA, can actually be thought of as houses or survival machines for RNA to live inside. To understand how RNA chains can interact with their environments, replicate, and evolve, we first need to understand the simple process of base pairing. Chains of RNA are made of nucleotides, small molecules that come in 4 different types labeled A, C, U, and G. The backbone atoms of a nucleotide, shown here as a yellow bar, can form strong chemical bonds with the backbone atoms of any other RNA nucleotide. This means that different chains can have completely different sequences from left to right. The parts we call the bases of nucleotides, the colored sections labeled A, C, U, or G, are attracted to other bases sort of like a magnet, but they're selective about who they will stick to. G selectively pairs with C, A selectively pairs with U. When bases find their matches and stick together, we call it base pairing. Researchers have found that with a little bit of assistance, base pairing allows chains of RNA to replicate and evolve. Here's how it works. When a long chain of RNA is suspended in cool water with high concentrations of free nucleotides, the chain can act as a template for its own replication. Nucleotides automatically base pair with their partners on the existing chain. If their backbone atoms form chemical bonds with each other, And by the way, this is the part that currently requires assistance from researchers. We're not yet sure how this would have happened in the wild. A complementary RNA strand is born, one with the exact inverse sequence of the original. If the water is then heated, paired bases lose their grip, allowing both chains to act as templates when the cycle repeats. The great thing about this process is that every other RNA chain produced is a copy of the original, but sometimes mutations slip in. This means that as chains compete for survival and reproduction, true evolution, descent with modification, acted upon by selection, can operate on chains of RNA. As amazing as replication is, base pairing also gives RNA chains a second special ability. When placed in water cool enough for base pairing, but without enough free nucleotides for replication, chains will fold up and base pair with themselves. The end result is a complex shape with certain sticky bases pointing outward because they weren't able to find partners. These sticky, outward-facing bases can cause unique chemical reactions by interacting with other molecules in their environment. A folded chain of RNA capable of guiding a specific chemical reaction is what we call a ribozyme. Some ribozymes break certain molecules apart, others join certain molecules together. A ribozyme's specific function is determined by its specific shape, and its shape is determined by its sequence. If a mutation changes a ribozyme's sequence, the shape can be modified, and so can its function. When ribozymes were first discovered, scientists wondered how difficult it would be for random chains of RNA to evolve legitimate survival functions. Imagine, for example, a ribozyme that could build nucleotides out of molecules it finds in its environment. Across multiple generations, natural selection could promote and refine this ribozyme because the chain would tend to have access to more free nucleotides than its rivals, allowing it to replicate more often. To explore this idea, researchers at Simon Fraser University produced a large group of random RNA chains and examined them to see if any happened to be able to make nucleotides. Surprisingly, some actually could, but they weren't very efficient. Researchers selected out the successful chains and then used a lab technique called PCR to quickly replicate them with slight random mutations. After just 10 rounds of PCR, followed by selection, highly efficient nucleotide-building ribozymes evolved. These are molecules with the lifelike ability to actively participate in their own survival. These ribozymes, and many others produced through similar experiments, are beginning to blur the line between living things and simple chemistry. So to sum things up, the RNA world hypothesis is the simple idea that the first things to replicate and evolve on our planet may have been chains of RNA or something similar to them. While the basic idea of the RNA world does seem to give us a promising pathway to the origin of life, it's still very much a work in progress. As mentioned, one of several unsolved problems is, how did nature get backbone binding to function without the special enzymes or lab techniques we use today? While many researchers continue to focus on RNA, others are investigating alternative molecules, chemical systems that might replicate and evolve without assistance and could have given rise to RNA. Continual breakthroughs are being found in both avenues of research. I'm John Perry, and that's the RNA world hypothesis, stated clearly. This video was funded by the Center for Chemical Evolution, the National Science Foundation, and NASA. Though we do receive grants from time to time, Stated Clearly is made possible with financial contributions from viewers like you. To support us, visit our website at statedclearly.com and click Contribute. I'm happy to announce that you can now also support us on patreon.com. So long for now, stay curious.
This animation shows the process of RNA replication being carried out by a theoretical RNA replicase ribozyme. The replicase attaches to the end of the template strand. As the replicase moves along the template, it catalyzes the polymerization of a complementary strand. At a sufficiently high temperature, the individual strands of an RNA duplex will separate from each other. Upon cooling, the single-stranded RNAs may then undergo RNA folding. For each duplex, only one strand will form a functional ribozyme replicase. This newly formed replicase can use its complementary strand as a template to create yet more replicases. The following animation is meant as an analogy to explain the need for compartmentalization in the RNA world. Here, the RNA replicase is represented by a blue cube. If the replicase encounters another replicase, it can make a copy, resulting in two identical replicases. Our replicase, however, is not so picky. If it encounters an RNA that isn't a replicase, it will copy that too. In an environment where there are many different RNAs, the replicase will quickly become outnumbered. What's the solution? By placing the replicases in a compartment, such as a vesicle, the replicases are separated from the other RNAs and will only make copies of each other. This animation shows the process of protocell growth and division. RNA nucleotides shown as blue dots traverse the protocell membrane and are used by an RNA replicase to make a copy of another RNA replicase. Meanwhile, the protocell membrane is constantly growing through the addition of micelles. Increasing the surface area faster than the volume causes the protocell shape to become elongated and unstable. The protocell eventually splits into daughter protocells, with the contents of the original protocell randomly divided between the daughters.